In a soundproofed basement at Tel Aviv University, a team of scientists stopped watering a tomato plant and walked away. Nothing appeared to happen. The plant did not move, did not wilt visibly, did not do anything a human eye could register as distress. But the microphones stayed on. And what those microphones caught, once researchers finally slowed the recording down into a range human ears could process, sounded like a string of tiny, rapid pops, almost like bubble wrap being pressed in a steady rhythm. The plant was clicking, not occasionally, not randomly. It was clicking up to 35 times an hour in a frequency range between roughly 20 and 100 kilohertz, 4 to 5 times higher than the upper limit of human hearing. A healthy, unstressed plant sitting in the same room produced fewer than 1 click an hour. This was not folklore. It was not a metaphor about nature being alive in some poetic sense. It was a measurable, repeatable, peer-reviewed finding, published in the journal Cell in March 2023, and it opened a genuinely new field in biology. The question of whether plants are, in some functional sense, talking, and whether machines could finally learn to listen. For most of the history of modern biology, the working assumption was straightforward. Plants were understood as reactive organisms, capable of responding to light, gravity, and touch through slow chemical and hydraulic processes, but not as organisms that actively broadcast information about themselves and to their surroundings. Scientists already knew plants released volatile organic compounds when damaged, chemical signals that drift through the air and can trigger defensive responses in nearby plants of the same or related species. That much had been documented for decades. What remained an open and mostly unexplored question was whether plants also produced sound in the way animals under stress often do, and whether that sound traveled far enough through open air to matter to anything listening. The researchers behind the discovery, led by Itzhak Kate and senior scientists Lilik Hadani and Yossi Yovel, had a simple but stubborn question. Plant physiologists had known for decades that water columns inside a stressed plant's stem can develop tiny air bubbles, a process called cavitation and TAT. This process creates vibrations. What nobody had confirmed was whether those vibrations ever left the plant and became actual airborne sound that could travel through open air and be picked up at a distance by something else. To test it, the team built an isolated acoustic chamber, positioned ultrasonic microphones about 10 centimeters from tomato and tobacco plants, and then subjected the plants to two kinds of stress: drought, by withholding water for several days, and physical injury, by cutting the stem. They also recorded healthy control plants and empty pots of soil to rule out background noise. The results were unambiguous. Drought-stressed tomato plants produced around 35 clicks per hour. Drought-stressed tobacco plants produced around 11. Plants that had just been cut produced a different pattern again, roughly 25 clicks per hour for tomato and 15 for tobacco. Untreated plants, sitting quietly with full access to water, produced less than 1 click per hour on average. The sound pressure level measured about 65 decibels at a distance of 10 centimeters, which the researchers noted is comparable in loudness to an ordinary human conversation, and just pitched far above what we can perceive. At a distance of 3 to 5 meters, they calculated, these clicks would likely still be audible to any animal with ultrasonic hearing, which includes many species of moths, bats, mice, and other small mammals that already navigate the world by sound frequencies invisible to us. The next step was where artificial intelligence entered the picture. The researchers fed their recordings into a machine learning model and asked it to do something no human listener could reliably do: distinguish, from the acoustic pattern alone, whether a given plant was dehydrated, physically injured, or completely healthy. The model succeeded, correctly sorting the recordings into these categories at a rate well above random chance. In other words, the clicks were not just noise, they were structured. They carried information about the plant's specific internal condition, and a trained algorithm could decode which condition that was, using nothing but the sound. The team didn't stop at the acoustic chamber to make sure the effect wasn't some artifact of the sealed artificial environment. They repeated the recordings in a much noisier setting, a working greenhouse, while simultaneously tracking each plant's physiological measurements, things like stem water potential, to confirm the sound patterns actually tracked real internal stress rather than something incidental. The correlation held. Drought-stressed plants clicked more as their internal water deficit worsened, and the rate of clicking shifted again as the plants dried out further, suggesting the signal wasn't a single on or off switch, but something closer to a graded readout of the plant's condition over time. This is part of why the paper was able to clear the bar for publication in Cell, one of the more selective and heavily scrutinized journals in the life sciences, rather than remaining a preprint curiosity. It's also worth being precise about what the acoustic mechanism itself is understood to be. The leading physiological explanation is a process called cavitation. Water inside a plant's xylem, the tissue that transports water from roots to leaves, is normally held under tension, almost like water being pulled up a very thin straw. Under drought stress, that tension can become severe enough that tiny air bubbles form and collapse rapidly inside the water column. And each of those micro collapses releases a small burst of mechanical energy, registering as an ultrasonic click. This means the sound is very likely a physical byproduct of the plant's internal plumbing failing under strain, not a deliberate vocalization in any sense comparable to an animal call. The word scream, which shows up constantly in headlines about this research, is a journalistic shorthand, not a term that scientists themselves use to describe the underlying mechanism. What makes the finding significant isn't that the plant intended to communicate distress. It's that the resulting sound happens to be structured enough and loud enough that it could plausibly carry usable information to anything nearby capable of hearing it. This is the part worth sitting with for a moment, because it reframes something we've walked past our whole lives without noticing. Every field, every greenhouse, every row of crops under stress has apparently been broadcasting information about its condition continuously for as long as plants have existed, in a frequency range we simply never evolved the biology to detect. As Professor Hadani put it in describing the work, even in a quiet field there are sounds we don't hear, and those sounds carry information. The plant kingdom wasn't silent, we were deaf to it. Machine learning is the first tool humanity has ever had capable of closing that gap. The obvious next question is whether anything in nature was already listening. That question has since been tested directly. A follow-up study from the same broader Tel Aviv research community focused on the moth species Spodoptera littoralis, examined how female moths decide where to lay their eggs. Moths in this genus rely heavily on hearing, since many of them have evolved ultrasonic sensitivity, specifically to detect the echolocation calls of predatory bats. Researchers gave female moths a choice between plants and recorded their oviposition behavior in the presence of the ultrasonic clicks typically produced by drought-stressed tomato plants. The moths incorporated those acoustic cues into their decision-making, adjusting where they chose to lay their eggs based on the sound signals coming from the plant. This was published as the first documented evidence of direct airborne acoustic interaction between a plant and an animal, meaning a case where an animal appears to base a real survival decision, in this instance where to place its offspring, partly on sound a plant is producing. The logic behind why a moth would care is fairly intuitive once you think about what oviposition actually risks. A female moth laying eggs on a drought-stressed plant is placing the next generation on a food source that may not survive long enough to support the larvae through their full development. A plant broadcasting acoustic evidence of its own water stress, even unintentionally, would functionally be handing a listening insect exactly the information it needs to avoid that outcome. The researchers behind this study now suspect this may not be limited to one moth species. They've raised the possibility that other insects relying on ultrasonic hearing, including some pollinators, could plausibly use similar drought-related cues when deciding where to forage, though that broader claim is still a hypothesis awaiting its own dedicated testing rather than a confirmed finding. It's worth being precise about what this does and does not establish. It does not prove plants are deliberately signaling in the way animals communicate. The leading physiological explanation for the clicking itself is mechanical, a byproduct of cavitation in the plant's water transport system, not a nervous system choosing to vocalize, but it does establish that the sound is functional information, real enough that at least one insect species appears to have evolved the capacity to use it. Once scientists had proof that individual plants were emitting readable signals, attention naturally turned underground, to a much older and more contested idea: the theory that forests are interconnected through fungal networks capable of sharing resources and warnings between trees. This concept, often nicknamed the Wood Wide Web, describes something called a common mycorrhizal network, formed when the thread-like filaments of mycorrhizal fungi, called hyphae, physically link the root systems of multiple plants, sometimes across different species. The underlying biology here is not in dispute. Mycorrhizal symbiosis is one of the oldest and best documented relationships in botany. Fungi extract sugars from plant teas in exchange for water and soil nutrients the plant cannot easily reach on its own, and in laboratory in some field conditions, measurable transfer of carbon between connected plants through these fungal bridges has been demonstrated since the 1990s. Where the science gets genuinely contested is in the leap from that established biology to the much more dramatic popular narrative. The idea that mature trees deliberately funnel resources to their own offspring through these networks, or that trees send chemical alarm signals to warn distant neighbors of insect attacks, coordinating a kind of forest-wide defense system. In 2023, a team of mycorrhizal ecologists, Justine Karst, Melanie Jones, and Jason Huxma, published a widely discussed review in the journal Nature, Ecology, and Evolution examining exactly these claims. Their conclusion was blunt. They found no peer-reviewed, published field evidence supporting the specific claim that mature trees preferentially send warning signals or resources to their own seedlings through these networks. They also found that the broader claim of these networks being widespread and structurally mapped across real forests rested on only a small number of studies, mostly from a limited range of forest types, and that citations across the scientific literature had progressively overstated what those original studies actually showed. Suzanne Simard, the ecologist whose research popularized much of this narrative, has defended her work and disputes some of the review's framing, and the scientific conversation on this topic is still actively unresolved. Part of what makes this debate worth understanding, rather than skipping past, is how it happened. Karst and her co-authors didn't just re-examine the original field data, they traced how those original studies had been cited and recited across hundreds of later papers and popular articles, and found that the descriptions kept drifting further from what the source studies had actually measured, growing more dramatic with each retelling, the way a detail shifts in a game of telephone. A single foundational study from a Douglas fir forest in western Canada, for instance, is now regularly cited as general evidence that trees transfer nutrients to each other through fungal networks, even though the original study wasn't designed to test that claim directly. None of this means the underlying biology is fake. Mycorrhizal fungi connecting to multiple plants at once is well documented, and further research specifically focused on disease signaling, including a 2025 study showing that plants infected with a fungal pathogen can transfer a defense-triggering compound called jasmonic acid to healthy neighbors through a shared fungal network, has continued to find real evidence for at least some forms of chemical transfer through these connections. The honest summary is this: the fungal connections are real, the potential for some communication and resource sharing is real and biologically plausible and supported by specific, documented cases. But the more cinematic version of trees actively parenting their young or issuing coordinated forest-wide alarms through this network has significantly outrun what has actually been proven in the field, and the scientists who study this most closely are the first to say so. What is not contested and does not require any romantic reframing is that plants use these same underground fungal channels for open chemical warfare. Black walnut trees are the textbook example. Their roots release a compound called juglone into the surrounding soil, and juglone is directly toxic to many other plant species, suppressing the growth of anything trying to compete nearby for light, water, and nutrients. This is not a modern discovery, and it did not require artificial intelligence to detect. Farmers and gardeners have known for generations that almost nothing thrives directly beneath a mature black walnut. What recent research adds is more precise mapping of how selectively these compounds spread, showing that the plant's chemical output is not indiscriminate poison, but something closer to a targeted competitive strategy, sparing its own symbiotic fungal partners while suppressing rival plant species nearby. A separate and genuinely striking line of research, this one not dependent on AI at all but on patient direct observation, comes from Yu Fukusawa's laboratory at Tohoku University in Japan. In a study published in the journal Fungal Ecology in September 2024, Fukusawa's team grew a wood, decaying fungal species called Phanerochaeta velutina around small wood blocks arranged in two different geometric patterns, a circle and a cross. If the fungal mycelium were expanding randomly with no sensitivity to the layout, it should have spread outward in roughly the same pattern regardless of the block arrangement. That is not what happened. In the circular arrangement, the network grew dense, connective webino, G around the outer blocks, but largely avoided expanding into the empty center, as though it had determined there was nothing worth reaching there. In the cross arrangement, the network built up unusually dense connections at the four outer arms, treating them almost like forward outposts for further foraging. Fukusawa has been direct about what this does and doesn't mean. He describes it as a form of memory and decision-making, built entirely from the coordinated behavior of thousands of individual hyphal tips, sensing local conditions and interacting with each other, with no brain, no central nervous system, and no single controlling structure anywhere in the organism. It's a legitimate, published, and fairly remarkable finding about the outer edges of what we're willing to call cognition. It is worth being clear that this experiment did not rely on AI to interpret the rhyme. Fungus. The intelligence being described here is the fungal network's own behavior, not a machine learning system decoding it. Fukusawa's earlier work, going back to 2019, had already shown something in a similar spirit using a different setup. A mycelial network was allowed to grow from a Starton G piece of wood toward a new food source, and once it successfully colonized that new resource, the original starting material was physically removed and placed in fresh soil elsewhere. Even disconnected from the network it had built, regrowth from that original piece of wood was consistently stronger on the side that had originally faced the new food source, as if some form of directional memory had been retained at the tissue level, despite the fungus having no centralized nervous system in which to store it. Researchers studying this describe several plausible mechanisms for how a brainless organism might hold onto information like this, ranging from short-term memory stored in the physical structure of individual growing hyphal tips to longer-lasting changes in gene activity across the network. None of these mechanisms resemble memory the way a human brain stores it, but functionally, in terms of the organism's later behavior, the network was acting on information from an earlier point in time. Now to the part of this story where artificial intelligence returns to center stage, and where the science is newer, thinner, and considerably more provisional than the plant acoustics research. Since around 2020, a research program led primarily by Peter Glor, working across MIT, the University of Cologne, and several smaller labs, has been investigating whether plants generate measurable electrical responses to specific human beings. The underlying biology is plausible enough. Plants do carry electrical signals generated by shifts in membrane potential and ion movement across cells, and these signals are known to change in response to light, touch, temperature, and injury. The Glor group's experiments attach simple electrodes to the leaves and soil of individual plants, most often basil, tomato, lettuce, or a houseplant called Tradescantia pallida, and record how the plant's voltage output shifts when specific people move nearby, speak, or perform practiced physical gestures near it. A random forest classifier applied to this data was reportedly able to distinguish between the presence and absence of nearby human movement with about 63% accuracy in a November 2025 study, a modest but statistically real improvement over the 50% you'd expect from pure chance. The same body of work reports being able to distinguish which of two specific individuals was near the plant with around 66% accuracy, and in a separate experiment, using a deep learning architecture called ResNet 50 to analyze plant voltage data converted into visual spectrograms, the system reportedly classified human emotional states with accuracy as high as 97%. That last number sounds extraordinary, and it deserves a proper amount of caution rather than pure amazement. This research has not gone through the kind of large-scale, multi-lab replication that the Tel Aviv acoustics work has, and several of these studies are explicit about their own limitations. Single plant setups, comparatively small sample sizes, and reliance on automated facial expression. Software rather than confirmed internal emotional states to generate the labels the AI was trained on. One related pilot study using the same general approach, published separately in late 2025, reported considerably more modest numbers. 85% accuracy detecting whether a lamp was on or off based on a plant's bioelectric signal, which is a fairly straightforward environmental sensing task, but only 73%. T accuracy distinguishing happy from sad emotional states using a single Purple Heart houseplant in fewer than 2,000 recorded samples. The researchers themselves described their emotion classification findings as preliminary and exploratory rather than established fact, explicitly stating that the results demonstrate feasibility rather than definitive evidence of plant-based emotion sensing, and they call for replication across different plants, locations, and research teams before anyone should treat this as settled science. It's also worth noting that this entire strand of research sits outside mainstream plant biology. The lead investigator is a specialist in organizational information systems and network analysis, not a plant physiologist, and the work has mostly appeared in smaller specialty journals and preprint servers rather than the flagship biology journals that publish the Tel Aviv Acoustics and Tohoku Fungal Studies. That doesn't make the findings false, but it does mean the appropriate level of confidence here is considerably lower than the confidence warranted by peer-reviewed, independently replicated work like the plant sound research. The honest way to present this line of research is as a genuinely interesting and still open hypothesis, not as proof that your houseplant knows how you're feeling. What is more modestly and more solidly supported, even in this newer body of work, is the simpler and less dramatic underlying idea that plants register environmental changes, including the presence of nearby movement, through their own bioelectric activity, and that this sensitivity may plausibly have evolved as an early warning mechanism against large herbivores approaching before contact is made. Put the sturdier findings from these separate lines of research next to each other, and a coherent, evidence-backed picture does emerge, even without leaning on the shakier claims. Plants under drought or physical injury produce structured, informative ultrasonic sound, verified in a peer-reviewed cell paper and independently reproducible. At least one insect species incorporates that sound into a real survival decision, verified in reviewed research from the same broader lab. Community. Fungal networks connecting plant roots are real, carbon transfer through them has been measured, and some fungal networks demonstrably alter their growth strategy based on the spatial arrangement of resources around them, all independently documented and largely uncontested. And what remains genuinely unresolved is how far this goes, whether trees are running anything like system through those fungal links, and whether plants are picking up on individual human identity or emotional states in any reliable, repeatable way. Good science means being willing to say that The exciting headline and the actual data don't always match up yet, and that some of what circulates as settled fact about plant intelligence is still, honestly, an open hypothesis waiting on better evidence. There's a genuinely practical payoff sitting inside the solid part of this research, and it has nothing to do with mysticism. If a machine learning model can already tell, from ultrasonic sound alone, whether a tomato plant is dehydrated before any visible wilting appears, that capability has direct value for agriculture. Hadani and her colleagues have pointed to exactly this application, imagining acoustic sensors placed in fields and greenhouses that listen continuously to crops and flag water stress in specific rows before yield is affected, allowing irrigation to be targeted rather than applied on a fixed schedule across an entire field. Given how much of global agricultural water use goes toward fixed schedule irrigation, regardless of actual plant need, even a modest improvement in this kind of early detection could meaningfully reduce water waste at scale. This is one of the more grounded end-ear term outcomes of the entire field, and more useful in the short run than any question about plant consciousness or plant emotion. It's also a natural extension of a trend that was already well underway in agricultural technology before any of this acoustic research existed. Soil moisture sensors, infrared imaging to detect early canopy stress, and satellite-based vegetation indices are already standard tools on large commercial farms. What the Tel Aviv findings add to that toolkit isn't a replacement for those methods, but a new data channel entirely, one that appears to register stress earlier than visible wilting, and doesn't require line of sight to every plant the way a camera does. Whether ultrasonic listening actually outperforms existing sensor networks in real field conditions, at scale, over a full growing season, is still an open engineering question rather than a settled one, since the published research so far has largely taken place in controlled chamber and greenhouse conditions rather than sprawling commercia. Whole fields with wind noise, insect activity, and machinery in the background. That next step, translating a laboratory finding into a reliable commercial product, is typically where promising biological discoveries either prove their practical value or quietly stall out, and it's too early to say which path this one will take. Stepping back from the specific numbers, what this research area actually demonstrates is something narrower and, in some ways, more interesting than the most viral claims about it. It is not proof that plants think, feel, or possess anything resembling awareness in the way animals do. None of the peer-reviewed literature makes that claim, and the researchers doing this work are consistently careful to say so themselves. What it does demonstrate is that plants are far more responsive to and communicative about their own physical condition than the traditional picture of a passive, purely photosynthetic organism ever allowed for. They generate detectable signals when stressed. Those signals carry decodable information. At least some of that information appears to be used by other organisms in the environment. And for the first time, we have tools, specifically machine learning models, trained to find patterns in signals too fast, too high-pitched, or too subtle for a human observer to parse directly, that let us listen in on part of that exchange. What we do with that capability is still an open question, and it's one worth ending on honestly rather than dramatically. The clearest, best-supported use case right now is agricultural. Catching crop stress earlier, using water more efficiently, building sensor networks that let farmers respond to what a field is actually experiencing rather than a fixed calendar. The more speculative applications, from decoding tree to tree, warning networks to reading human emotional states through a houseplant's wiring, remain active, legitimate areas of ongoing research. But they are not yet established science, and treating them as though they were does a disservice to the genuinely solid findings sitting right next to them. The tomato plant in that soundproofed room in Tel Aviv really was producing structured, informative signals in a range no human could hear, and a machine really did learn to decode what those signals meant. That part is real, published, and repeatable. Everything built on top of it deserves the same standard before we accept it as fact. If this kind of deep dive into what the research actually says, separating the solid findings from the speculation, is useful to you, subscribe for more breakdowns like this one, and let me know in the comments which scientific claim you'd like fact-checked next.